Right, the formation of pseudopodium, for example, we take the amoeba, formation of pseudopodium, different types of theories will be there. Allen's theory and Pontanian theory will be there. Let us see how the pseudopodium is formed in the amoeba. For example, this is we take the amoeba. Okay, it is nucleus, it is vacuoles. Here basically two types of cytoplasm we are seeing. First is goes to inner side. There is have a liquid shape, liquid type of substance, liquid type of cytoplasm we call as sol, and the outermost part we consideration as a gel. Okay, here sol is converted to the gel, gel is converted to the sol. In these conversions means vice versa. Sol is converted to the gel, gel is converted to the sol, vice versa, to project towards to formation of a pseudopodium to move one place to another place. This type of movement we call as amoeboid movement. How the formation means, for example, this is a sol to elimination of water molecules, formation of gel. The process we called as gelation. Gel to require to taking water to formation of a sol. This part is goes to solation centers. Okay, gelation and solations. To vice versa converts it to the here sol to elimination of water molecules to come outwards to formation of a gel to produce the formation of pseudopodium. And last one here is goes to the sol to elimination of water molecules to formation of a gel. Like this vice versa is conversion will be takes place, the pseudopodium will be formed. These type of slow movement we call as amoeboid movement. In the higher organisms also this type of movement by the WBC will be exposed to this type of amoeboid movement. Okay, this type of amoeboid movement will be there. This is about pseudopodium. Next we are going to discuss about flagella. Next we goes to flagella. Flagella, another type of locomotor organelles in the protozoans, mostly mastigophores. We called as flagella. Mastigo means some small hair-like projections we call as mastigophoridae means having and bearded, we call as mastigophores. Basically it is a whip-like structure. It is goes to whip-like structure, and it is originated from the anterior part of the organisms. It is a formed anterior part of organism, that is a flagella. That is the typical flagella. There is have a different type of structures. First we called as go to ultrastructure of flagella. The first one is goes to here is goes to axoneme. Axoneme covered by the small membrane we called as plasma membrane. Okay, first one is goes to ultrastructure of flagella. First one is goes to axoneme. Axoneme is covered by the plasma membrane. Outermost we called as plasma membrane. Okay, inner complete this space we called as a matrix. What are the flagellar organelles? What are the parts of the flagella? It is embedded in the matrix. It's complete we called as matrix. In the entire the round structure of the matrix, all the cell organelles and what are the components of flagella is embedded into this type of axoneme. Second one is goes to microtubules. Microtubules, there are two types of microtubules are there. For the center, there are two microtubules are there. That goes to central singlet units or central singlet microtubules are there. All the microtubules are made up of the protein is goes to tubulin. Tubulin is a protein is a formation of a microtubules. In the central there are 2 are there, and the peripheral there are 9 doublets are there. 9 doublets. 3, 4, 5, 6, 7, 8, 9. The 9 doublets are there. This type of arrangement is goes to 9 plus 2 type of arrangement. Basically, this type of flagella we are seeing naturally protozoa, and all eukaryotic flagella also bearing of same type of arrangement. But flagella we are seeing into bacteria. A bacteria flagella we should not possess to these 9 plus 2 arrangement. Only eukaryotic organisms, that is goes to protozoa, is possessed to the 9 plus 2 arrangement. And goes to spermatozoa also. Higher organisms, the spermatozoa also, the tail we possess to this type of 9 plus 2 arrangement will be there. Here we are seeing into this doublet. This doublet, this is goes to, first part is goes to outer pass doublet we called as A tubule. A tubule also we consider as alpha tubule. It is comes to the outer part. This outer part, it is smaller than the big one. Smaller than the B one. But innermost part we consider as a B tubule. B tubule, nothing but we call as beta tubule. Beta tubule is considered as a big one. A is a small one, B is a big one. And A is a complete one. A is a complete circle, B is not complete, it is incomplete circle. It's very clear for that. A is the outermost A tubule, B is the innermost. It is innermost is the B tubule. Okay, A nothing but because alpha tubule, B tubule we consider as a beta tubule B. A is a complete tubule, B is an incomplete tubule. Okay, each and every 9 peripheral tubules is interconnected by the small connections. These small connections we called as nexons. All connections we called as nexons. All connections we called as nexons connections. Here, next one is goes to A tubule to formation of a 2 arm-like structures. 2 arm-like structures here. These arm-like structures to be formation of clockwise directions. To A tubule is connected to the B tubule for the 2 arm-like structures. These arm-like structures we call as dynein arms. We call is called as dynein arms. These arms, dynein is protein is a formation of a dynein arms. Dynein arms to catch to the A from the A to B. It is catch to clockwise direction of orientation will be there. This type of arrangement is goes to peripheral tubules. Next is goes to in the central tubules is covered by the one sheet we called as inner sheet. We called as inner sheet. Inner sheet. Inner sheet. Next, outermost peripheral tubules is covered by the one more sheet we called as outer sheet. Outer sheet. Fourth one is goes to outer sheet. Outer sheet. Right, here one A tubule to be originated by the one spoke-like structures we called as radial spokes is similar to the wheel of bicycle. There is have a spokes. How the spokes is arranged to the rim to the central axles. These type of spokes we called as radial spokes. We called as radial spokes. Radial spokes will be there. It is try to attach it to the central filament or central tubules. Right, this is arrangement of ultrastructure of the flagella. Right, so I will tell you once again. First one is goes to a round structure of axoneme. In the center part, to complete the space we call as matrix. What are the cell organelles? What are the components of the flagella? It is embedded into the matrix. The matrix be consisting of a number of microtubules. The microtubules are goes to two types of microtubules are we are seeing. First one in the center, there are the two central singlets. Two central singlets tubules will be there. All the tubules are made up of tubulin is a protein. The center is goes to central two tubules are there. In the peripheral 9 doublets are there. Means 2 molecules, 2 tubules are embedded to 9 tubules will be there. This arrangement we call as 9 plus 2 arrangement. 9 plus 2 arrangement will be there. Okay, it is a peripheral. There are 2 doublets which consist of A tubule, outer one, B tubule and inner one. A and B. A is a complete circle, B is an incomplete circle. These each and every doublets to be arranged to the clockwise direction by attachment to the small connections we called as nexins. We called as nexins connections will be the nexins connections. And A, from the A, 2 arm-like projections to be formation. These arm-like projections we call as a dynein arms. The dynein arms is connected to the from the A2 is connected to the B like clockwise orientation will be the at here. And let us wise goes to the central tubule is covered by the one sheet which goes to inner sheet. All the peripheral tubules is covered by the one more sheet is goes to peripheral sheet the outer sheet. Next here the central tubule is connected to the peripheral tubule by the small spoke like structures we call as radial spokes will be there. This is about ultra structure of flagella.